So welcome back from the break. Let's take a look at our two concept check questions and then we'll move on. So give at least 4 examples of structural and functional diversity found in protists. Well, if we want to look at structural diversity, we've got unicellular versus colonial versus multicellular. There's a structural diversity right there. We could talk about the presence of unique organelles. So, for instance, the presence of contractile vacuoles in the ciliates not found anywhere else amongst eukaryotic cells. So there's 2 structural differences. If we want to talk about some functional differences, we could think in terms of photosynthetic, or let's say autotrophic, heterotrophic, and mixotrophic. Those are functional diversities. We could talk about the differences in locomotion. So you've got ciliated protists that use cilia or flagella, flagellated protists for locomotion. And then you've got protists that use amoeboid action. So the classic amoeba. There's some more functional differences and structural differences. The list goes on and on. It's basically open to how you want to interpret that. You just need to be able to justify including that by giving some examples of members of the protists that illustrate the ideas that you're putting forward here. Briefly describe the role of endosymbiosis in eukaryotic evolution. Well, first you have to think in terms of the primary endosymbiosis of mitochondria and subsequently chloroplasts, and then you think in terms of the secondary endosymbiosis of the 2 lines of resulting algae, the red algae and the green algae, to give rise to the broad diversity of photosynthetic protists that trace their chloroplasts back to either the red algae or the green algae. And then, of course, if you want to think about the chlorarachnophytes, you probably have to think in terms of the possibility of a second primary endosymbiosis, if not a different line or a later secondary endosymbiosis, certainly a much later primary endosymbiosis based on the molecular evidence. So endosymbiosis is one of those ideas that very neatly explains the broad diversity that we see amongst the protists in terms of photosynthetic and non-photosynthetic lines. So it's kind of important. So let's take a look at the first clade, known as the excavator. And the reason why they're called the excavator is because if you look at the surface of the cell, somewhere on the surface of that cell, there is a large groove or pocket that is formed by the reorientation of the cytoskeleton within the cell. And all members of the excavator have this groove or pocket somewhere on the surface of their cell. And so it's characteristic. Of course, it is supported at the gene level by genetic sequencing. So we are fairly confident that this group does represent a clade. The controversy lies specifically with the euglenids because of the mixotrophs that are found amongst the euglenids. Because you've got these cells that can be both heterotrophic and photosynthetic at the same time. And that trying to explain that origin is somewhat controversial. And then amongst the euglenozoans, you've got a whole line of parasitic members of the euglenozoans, and then amongst the diplomonads and the parabasalids, and particularly the diplomonads and the parabasalids, both have what are referred to as reduced mitochondria. And at one point, they were actually considered to be the most ancient lines of eukaryotic cells because they were considered to not have had mitochondria. But that has now changed. So there's a little bit of controversy surrounding this group because of the members that are in it. But in terms of it being a supergroup, the molecular data suggests very strongly that this is a supportable supergroup. So let's look at the individual members. So as I said, the diplomonads and the parabasalids are interesting because at one time they were thought to represent the oldest of eukaryotic cells because they lack functional mitochondria in the sense that we think of functional mitochondria. In other words, there are vestigial, I guess, mitochondria, or to use the term that seems to be current, reduced mitochondria, in that they do not carry out the aerobic portion of cell respiration. These are organisms that are strictly anaerobic, and the reason why is because they are parasites. They don't need to do aerobic respiration. They can get their energy sources from the organisms that they parasitize. And so in the usual way in evolution, if it costs more to maintain a structure than the benefit you get from it, you discard it. So it's not a use it or lose it. It's that you could maintain it and you could use it, but because you have alternative ways of getting the product that you would get from the structure, cost benefit says use the cheaper way of getting it, using the host in this case, and discard the more expensive way, maintaining a mitochondria that requires an input of resources or a utilization of resources that could otherwise be used somewhere else. So it's not use it or lose it, as Lamarck would have had it. It is very much a cost-benefit analysis. How much does it cost you to retain it versus how much benefit do you get out of it? Is there a cheaper way of getting the same benefit? And in the case of these parasitic organisms, there most definitely is. So let's look at the diplomonads first, because they are also interesting for another reason. So even though they lack functional mitochondria, we know that they had mitochondria once, and the evidence is very clear cut. If you look at the genomic DNA, the DNA in the nucleus, you find the same mitochondrial genes that are the product of horizontal gene transfer from the bacterial precursor as are found in all other eukaryotic cells. Those genes are present in the nucleus of diplomonads, and let me add in there, in the nuclear DNA of the parabasalids as well. So we know that at one point, both diplomonads and parabasalids really did have functional mitochondria, but they've discarded those functional mitochondria because they just don't need them anymore. They are parasitic, so they can tap into the energy resources of their hosts. Plus, the environments that they generally live in are anaerobic, very little oxygen, so there would be no point in doing aerobic respiration anyway, because you don't have the oxygen to support it. What makes diplomonads really interesting is the fact that they give a suggestion as how evolution may have transitioned from the haploid condition of prokaryotes to the diploid condition of eukaryotes. Because somehow you have to be able to explain the origin of a diploid nucleus. We can explain the presence of the nuclear envelope fairly easily by hypothesizing invagination of a cell membrane, a plasma membrane, to form a membrane-bound organelle around the DNA, the developing quote-unquote chromosomes. But how do you go from a haploid condition to a diploid condition? Well, the diplomonads give us at least a potential solution, or at least a stepping stone along the way, because they contain and maintain throughout the life of the organism two separate haploid nuclei. So it's not just two equal-sized nuclei, but two equal-sized haploid nuclei. So functionally, they are diploid, but they are diploid because they have two haploid nuclei. So that gives us at least a potential hypothesis to try and explain the origin of a diploid nucleus. So you start out with a haploid structure. You get duplication of that as it develops into a nucleus. So you have two haploid nuclei. Eventually, somewhere along the evolutionary timeline, it becomes more efficient to fuse those two nuclei together to create a single diploid nucleus, which is something that the vast majority of eukaryotic cells continue to utilize to this day. It is more energy efficient to do that. And it's more efficient in terms of cell division, transmission of chromosomes, everything that you can associate with efficiency as far as a eukaryotic cell is concerned, tends to favor a single diploid nucleus. So diplomonads are all parasites. The most infamous of the lot is Giardia intestinalis, which is a gut parasite of mammals, including humans. You typically get infected by drinking contaminated water. This is one of the reasons why you should never drink from a stream in the continental US and perhaps anywhere in the world at this point, because this is a very widespread parasite, and it is found in most quote unquote natural streams. You really do need to sterilize the water in some fashion, either with chlorine pills or by boiling, because if you get one of these infections, you will wish you hadn't, because it gives you diarrhea, intestinal upset. It's pretty discomforting to have it. Fortunately, there are medications that can cure it, but it takes about a week to week and a half to get rid of the infection. And during that time, you feel particularly uncomfortable. The parabasalids, oh, well, this is a photograph of Giardia. So you can see the groove, the pocket on the surface of the cell. Also note many flagella. So it's a multiflagellated cell. And remember, these are eukaryotic flagella, not prokaryotic flagella. So these are distinctly different from the flagella that we saw in the previous chapter associated with bacteria. So these are the flagella that are made out of microtubules. They're an extension of the cytoskeleton. They have that classic 9+2 layout of microtubule structures, a pair of microtubules running up the center, that's the 2, and then 9 pairs of microtubules around the rim of the flagellum. And also we can say in here the cilia, because they share a similar internal structure. They're just different in length. And the microtubule pairs in the rim are connected by dynein arms, whose interaction with the microtubules set up the sine wave, the S wave in the flagella, or cause the ciliates to rock backwards and forwards in a sideways motion to provide locomotion. The parabasalids have a unique feature on the outside referred to as an undulating membrane. It's actually an extension of the plasma membrane. Embedded in that, you're going to find symbiotic bacteria. There is some question as to what actually provides the locomotion here. Is it the undulating membrane, because that can provide a means of locomotion? Is it the actual flagella of the parabasalids, or is it the symbiotic bacteria that are providing the means of locomotion? It's not clear. What we do know is that these are, again, all parasites. They also have the reduced mitochondria that lack the ability to carry out aerobic cell respiration, but we know that they are, or they were once, fully functional mitochondria, because again, the gene sequences associated with mitochondria that are found in the nucleus are found in the nucleus of these organisms as well. So they're there. So there were fully functional mitochondria at one point. These are, again, parasites of mammals. The most famous or infamous as far as humans are concerned is Trichomonas vaginalis, which is a parasite of the human reproductive tract. Now, it says here that it causes yeast infections in human females. This is not a yeast, but a lot of people refer to the infection as a yeast infection because there are true yeast infections. And when we get to fungi, I'll talk about the fungi that actually cause true yeast infections. A lot of people mistakenly refer to Trichomonas infections as yeast infections. And the reason why is because these are opportunistic when the pH conditions in the female reproductive tract get out of alignment. That allows Trichomonas to infect the reproductive tract, and it often leads to the production of a rather smelly discharge, which is symptomatic of the presence of Trichomonas. Of course, it's also symptomatic of other vaginal infections as well, including true yeast infections. What is problematic and what a lot of people don't realize is that Trichomonas can also infect the urethra of the penis. And so males can also get infected with Trichomonas. But in the vast majority of male infections, it is cryptic, meaning there are no external symptoms to indicate a Trichomonas infection. So a male may be Trichomonas positive and pass it on to his sexual partners, not realizing that he has an infection. This is where the practice of safe sex, where knowing your sex partners becomes really important, because many of these STDs can be transferred from partner to partner because without anyone realizing it, because there are frequently no symptoms. Even in women, it's only in about 50% of all infections that you actually have the external evidence in the form of that discharge. Sometimes, again, it can be cryptic, no external symptoms. So the practice of safe sex and knowing your sexual partners clearly has some value under these circumstances. So here you can see Trichomonas. You see what's that extension of the cell membrane, the undulating membrane, some flagella. And remember, in the undulating membrane is where you find the symbiotic bacteria that are suspected to aid in the locomotion of the parasite. These are bacteria that are normal inhabitants of the vaginal tract in the case of Trichomonas. And some researchers have suggested that perhaps it was the symbiotic bacteria that in some fashion began the evolution of eukaryotic flagella. Not entirely sure how that took place. I'm not up to speed on the literature. I just know that this has been suggested. I'm not sure that I necessarily buy into that, but I think I would need to read the literature to know a little bit more about that. Then we have the euglenozoans, which are also excavator. So the classic euglenozoan that you would all be familiar with is euglena itself, and it has a similar structure to this. What is characteristic of all euglena is the pair, or is the presence of a pair of flagella. They're biflagellated. In fact, multiflagella, or at least biflagellation, is characteristic of the vast majority of flagellated protists. There is one supergroup that is unique in that this is not the case. And if you think about the names of the supergroups, you may be able to pick out which one it is, but I'll identify it for you when we get there later in the chapter. Within the flagella, you have these crystalline rods of protein that sit just above the basal body at the base of the flagellum. And here you can see that 9+2 structure of the flagella that I was talking about. We have no real clear idea what the function of this protein crystal structure actually is, but it's always associated with the flagella. So it probably has some role either in the formation of the flagella or in the action of the flagella. Not entirely sure what the situation is with this. So the first group of euglenozoans are the kinetoplastids. These are all, without exception, parasitic. They do have a functional mitochondrion, a single one that contains a mass of DNA called the kinetoplast. And that kinetoplast is characteristic of all these parasitic protists. And because it is a common feature of all these parasitic protists, it is thought that this kinetoplast may represent a way of targeting those kinetoplasts that are parasites of humans and give rise to a number of very serious diseases. So one disease that is caused by kinetoplasts is sleeping sickness, which is found in Africa. Someone with this disease is in a torpid state. They appear to be asleep. They're not really asleep. Mentally, they're fully alert, but their body is just in a torpid state. So they look like they're asleep. And this is lethal. And there is, as best I know, no known treatment for this. The parasite is carried by the tsetse fly, which is spelled T-S-E-T-S-E, but it's pronounced tsetse. And this was a plague when Europeans first entered Africa, because European livestock and Europeans themselves were very sensitive to the bite of the tsetse fly and diseases transmitted by it. And this is one of those diseases that was transmitted by it. And a lot of time and effort was spent trying to figure out ways, particularly in terms of cattle, to breed European cattle with native African cattle to obtain a line of cattle that were resistant to the tsetse fly, and specifically to the diseases that it carried, because the native cattle obviously had an immunity to the diseases that the tsetse fly was carrying. Another disease that is caused by the kinetoplast is Chagas disease, which is found in South America. And then a third one that has a more worldwide distribution is something called leishmaniasis, which is also caused by one of these kinetoplast parasites. And here you can see the parasites, these little ribbon-like cells adjacent to human red blood cells. So a human red blood cell is quite tiny, and so these parasites are very tiny as well, as you can see. Then we have the euglenids, which are the euglenas proper. These are the ones that you're most familiar with. Again, remember, these are biflagellated, but if you've ever seen euglena under the microscope, and hopefully you have, you'll probably recognize that there is only a single flagellum that is visible. That's because the second flagellum is typically truncated and never actually extends out of the flagella pocket at the posterior end of the cell. They also have a unique glucose polymer called paramylon starch. I have yet to be able to figure out exactly what makes it unique. When I've looked up paramylon starch in Google or other search indexes, I get the answer that it is the form of starch that euglenas use to store glucose, which is exceptionally helpful. And I've not been able to find any other really good source of information that tells me anything about what paramylon starch really is, what makes it different from starch in general. So whatever it is unique to euglena. So there's a protist unique feature. And again, as I mentioned earlier, there are also species within the euglenids that can be autotrophic. There are others that are heterotrophic, and then you have a few that are mixotrophic, and these are the ones that flip backwards and forwards between heterotrophism and autotrophism, depending on the light regime. So here is euglena. Hopefully you recognize this, and you can see what I mean in this artist representation about the truncated flagellum. So you have the flagellum that actually provides the locomotion, but then you have this truncated flagellum also in the pocket that the full length of the flagellum extends from. You have contractile vacuoles that allow it to expel water. It does have a kind of cell wall in the form of this pellicle, a polymer coat that wraps around it in a spiral fashion outside of the plasma membrane, has multiple chloroplasts scattered throughout the body of the cell, so it is truly photosynthetic. It has two light detecting organs. It has an eye spot, which provides directionality for the light and tells the cell which direction the light is coming from. And then it has a light detector that simply detects the presence or absence of light. So you've got these combined light detecting devices that provide both directional information and information about the presence or absence of light to the euglenas. So those are the excavator. So let's take another break. And here is the concept check question. And I'll meet you again on the other side of the break to answer this question. So the question is, why are the mitochondria of diplomonads and parabasalids often described as highly reduced? See you on the other side of the break with the answer.